Hello friends, today I am going to discuss on a topic that is gene symbol. This is a very interesting one because we know that all the genes, they need to be symbolized in order to study them or make it easier to analyze their functions or to map them on particular chromosome. And the need for this gene symboling was initially felt by Mendel himself. So while proposing his two laws on law of segregation and independent assortment, Mendel also used some of the alphabetic symbols to designate particular genes. Today we symbolize the genes keeping some basic principles or rules in mind. So in this lecture, my fundamental objective is to discuss with you regarding the rules and the principles that we adopt to symbolize the genes. Before starting the gene symboling principles, I would like to give a brief description of some of the terminologies that will help even a novice one to understand this gene symbol topic. The first one is character and trait. What do we mean by character and what do we mean by a trait? Now, a character is an attribute. It can be a physical attribute. It can be a biochemical attribute. It can be even a behavioral attribute, like the height of human beings, the skin color, coat color in rabbits, or anything you can think of. Now, these characters are generally identified on the basis of their different forms. Like for human beings, some are tall, some are short in height, some are having fair complexion, some are having darker complexion. In case of rabbit also, we find some albino rabbits with white fur color and some rabbits with gray fur color. So these alternative forms of the character is known as trait. So a character is an attribute and the different forms of the character is trait. Now, what Mendel hypothesized at that time of proposing the laws of segregation, that there are two terms again. One is phenotype and the second one is genotype. Now, the phenotype is the physical appearance of the character, how the character is appearing. So, a tall man is having a phenotype of tallness. A tall plant is having a phenotype of tallness. So, this is the physical appearance that you can say. And genotype is the genetic makeup that is determining the phenotype. I will come to the genotypic discussion later on, but for now, easy understanding that genotype is the genetic makeup that ultimately determines the phenotype, how the character will be expressed, which kind of a trait the character will express. Now, the third important thing that we need to understand before starting gene symboling is gene and allele. Now, Mendel hypothesized that for each and every character, there is a gene. So, there is a parallelism between a character and a gene. So, he hypothesized that each and every character is actually governed by a gene. Now, as a character is having alternative forms, a gene is also having alternative forms. These alternative forms, in case of character, it is known as traits. And suppose I make it trait 1 and trait 2. So a gene is also having alleles, or the alternative form. This is allele 1, allele 2. Now there was another parallelism that for a single character there is a gene involved and the different forms of the genes govern the different forms of the character. Means, suppose the trait 1, tallness, is governed by one allele, whereas dwarfness is governed by the second allele. So this is the basic principle of genes, how they govern the characters, and the alleles, different forms of the gene that express different forms of the character, that is trait. Now, Mendel also understood that there are two types of alleles actually. One is a dominant one and other one is a recessive one. From the name itself, it's very clear that a dominant one is powerful and recessive one is weak. So, from the first law of Mendel found that if I cross a tall plant with a dwarf plant, the resulting F1 plants become tall. So the two traits of the character of plant height is not equally expressed in the filial generation. So from that very finding, the essence of dominance and recessiveness was clear, but this recessiveness or the dwarfism is not totally absent, because if we allow this tall F1 plant to self-reproduce or self-fertilize, then again we get a 3:1 ratio of tallness and dwarfness. So the dwarf feature or the dwarf trait is somehow silent in this F1. So this trait is called the recessive trait, which cannot express itself, though it is present in the F1 hybrid, but upon its selfing in the F2 generation, filial 2 generation, this trait will again appear back. But the tallness is the trait that can express itself in the filial 1 generation. So this trait was termed dominant and this trait was termed recessive. So the dominant trait is governed by a dominant allele and the recessive trait is governed by a recessive allele. Now both the alleles are of the gene which is governing the character of plant height. So he used a common system to symbolize the dominant and recessive alleles of the same gene. So Mendel used, suppose capital A gene symbol, and proposed that suppose A gene is controlling the plant height. So A gene is now having two alleles. Written in capital letter is the dominant allele and written in recessive letter of the same alphabet, the recessive allele. So he proposed the monohybrid cross like this is tall and this is dwarf. Now this becomes capital A small a. Both the dominant allele and recessive allele is present, but phenotypically this plant is tall. So capital A is the dominant allele, whereas small a is the recessive allele. So with this basic information, we now move on that how gene symboling is practiced nowadays. So Mendel proposed that arbitrarily any alphabet can be taken to symbolize a particular gene. But this had a very strong limitation that that only 26 characters or 26 genes can be named, because we have only 26 alphabets in our hand. And they can have either capital or small forms for dominant and recessive alleles. Then when the subject of genetics was really expanding, we found several incidences where this gene symboling was not being able to be applied. Take the example of multiple alleles, where number of alleles of a particular gene is present, how they are going to be symbolized. So nowadays the gene symboling, we can really classify this gene symboling system into 3 or 4 categories. The first category is based on the dominant character. This is based on the dominant character. In this case, what we find that if I cross a tall plant with a dwarf plant, and if I get a tall plant, then it means that tallness is the dominant form or the dominant allele. So the gene will have the symbol T, capital T. And its recessive allele will have the symbol small t. Right? This is very simple. This is also even now it is followed in case of colorlessness or colorness of several traits in plants and animals. Like you can give the example of PR gene, which gives purple color to maize aileron layers. You can have the example of C gene, which is involved in regulating the coat color of rabbits. The colored rabbits are given the symbol capital C, whereas the colorless far rabbits are given the symbol of gene small c. So this is one derivation where the dominant trait or the dominant character's first letter is used as a symbol of the gene. Soon we come to understand that this system has a limitation. Because if we take the example of Drosophila eye color, we will find that in case of Drosophila, the dominant trait is a dull red color. So a dull red eye color is the dominant trait. But there can be several alternative forms of this character, like purple color, vermilion color, red color, and so on. So a single character is now having multiple forms of traits. So now how we are going to symbolize all of them? So a second derivation came up where the recessive trait is used to symbolize the gene. So we know nowadays that each and every eye color mutation in Drosophila is recessive in nature. So all the recessive mutations, suppose I say vermilion red color, then the gene symbol is given as V. Red eye color, the gene symbol is given as R. Or in this way. So what happens? The dominant allele becomes capital V, which gives you normal eye color, and small v, which gives vermilion eye color. The second one, R, gives you normal eye color, and small r gives you red eye color. So this V and R, both the genes, they're involved in governing the eye color of Drosophila, but their alleles, their different forms, do not lead to a similar type of phenotype. Means the small v allele gives you vermilion eye color, whereas the small r allele gives you the red eye color. So it's very apparent that this system is also lacking some of the key features, because we are not getting to know how many of the genes are actually involved in governing the normal eye color. So the third option of deriving gene symbol is mutant based. So in this case, a mutant allele is named. A gene is identified only through its mutation. For example, what is the function of a light or a bulb? If I want to know, then I will have to put the light off. Then and then only I can identify the real need of a bulb or the real role of the bulb. In a similar way, what is the role of the gene? It becomes apparent only from if I have a mutant of that gene where the gene function is disrupted. So in this way, there were two terms that was used. One is wild type and the second one is mutant type. Now, to be very precise, wild type means the trait or the form of the character which is most predominant in the population. Means if I go to a field of pea plants and I am surveying 10 lakhs of pea plants and suppose 9,99,000 plants are tall, it means that tallness is the wild type character, and the character which is inherited and a different from the tall like dwarf is the mutant character. So from this derivation, the name of the character which is governing the plant height will be named by the first letter of the mutant trait. So in this case, for tallness and dwarfness, the mutant trait is dwarfness. Now, the gene will be symbolized as D. Now there is a question that whether it will be small d or it will be capital D. Means the gene that is governing the plant height will be denoted by a small d or a capital D. Now this depends on the dominance relationship of this mutation. Means if I get a tall plant and I have got a dwarf plant. If I cross them, tall cross dwarf, and if I get a tall plant, this means that this mutation, dwarf mutation, cannot express itself in the F1 hybrid. So by definition, this is a recessive mutation. So if the mutation is recessive, then the gene symbol will be of small letter. Now, if I cross the same tall plant with a dwarf plant and I get a dwarf plant, it means that this dwarfness is no longer a recessive trait. Rather, it is a dominant one, as it can express itself in the F1 hybrid in heterozygous condition. So in that case, the gene symboling will be capital D. Now, how will I come to know that from the gene symbol itself that whether the capital D is the wild type or small d is the wild type? So I know that there is from the gene symbol itself, if someone gives me that suppose there is a gene called D. So I come to know that this is a mutant because from the point that of gene symboling, only a symbol will be given as per the mutant trait. So this is the dwarfness trait and recessive in nature. Now it can have, a population can have some dominant mutations also. So that dominant mutation may not be the wild type. Means this may be not the most frequent trait in the population, but still dominant. We know that most of the mutations are recessive in nature, but some of the mutations are really, like notched wing in case of Drosophila, is a dominant mutation. So in this case, the nomenclature will be capital N for this gene, the gene governing the notched wing. Now, to overcome this kind of a situation, how we can identify the wild type, another convention came out that designate the wild type by simply a plus sign. Now you think for a particular gene, the symboling may be plus D. This symboling gives me the idea that this D is a recessive mutation and it's a mutant, it's not a wild type. And another symbol can be plus N, which means notched wingness in Drosophila is a mutant, it's not a wild type character, it's not frequent in the population, but still the mutation is dominant in nature. So this is the most preferred gene symboling system. In some of the cases, we can use not the single N only, you can use NOT, means first 2 or 3 or some more letters of the gene, simply to increase the number of symbols that we can adopt. Because again, if we are on N only, then only a few gene symboling can be done. Suppose I have white and wrinkled 2 genes, both will be given the symbol W. But if I use WHT and WRK, then I can easily symbolize 2 other genes. One is white and another is governing the trait of wrinkledness. And with the advent of modern biotechnology, we have come to know a lot of gene sequences also. And we can now adopt reverse genetics. So genetics has 2 wings. One is forward genetics and the second one is the reverse genetics. In case of forward genetics, we know the phenotype first and we try to identify the genotype by analyzing the phenotype. In case of reverse genetics, we know the gene sequence, we know which kind of protein it can produce, and we can identify what happens if this gene is overexpressed or silenced. This kind of a study is known as reverse genetics, to know the function or the phenotype from the genotype. So as we know some of the sequence of the genes, a gene symboling can be done directly from the type of protein it is coding for. Like cyclins, CLN. You can name some genes as CDK, cyclin dependent kinases. Okay, so these are another way of gene symboling. So this is in a nutshell about the gene symboling system. But one thing I have not mentioned here, the case of multiple alleles. That what will happen in case of symboling genes which are having multiple alleles. A classical example is the far color in rabbits, right? So the colored gene was given the symbol C. Means this is the gene, the nomenclature was done on the basis of the dominant character, which is coloredness, and its recessive allele small c. This one governs the recessive trait, that is albinoness. Means the white rabbits that we find are actually recessive mutants. They lack the gene that produce color in their coat. Now, soon after the discovery of these albino rabbits, two other color variants were also observed. One is known as Himalayan and the second one is known as Chinchilla. They are also variants of color. Means the character is single. This is the color of coat for rabbits. But there are three traits now. One trait is total colorless albino. Second is Himalayan type. Third one is Chinchillan type. So how the gene symboling will work in this case? In this case, what we have done, that we name C as the gene because it is a dominant gene. A recessive mutation, small c, leads to total absence of pigmentation, gives albino, white far color. Now, in case of the Himalayan trait, we have assigned this allele C superscript H. Mean this is also a recessive mutation of the same gene, but it's a different allele. It doesn't give you totally albino rabbit, rather Himalayan type of rabbits. In a similar way, we name it CCH for the Chinchillan color. So this is the symboling that we adopt in case of multiple allelic series, where a single gene, you can also argue of symbolizing it as plus C, CH, CCH. So these are the gene symbols that govern the colors, where plus is the wild type, the dominant one. Small c is the recessive mutation that gives you albino. Another mutation of the same wild type, CH, now CCH. Now, it may be that several genes are involved in determining the same trait. One lecture that I will be taking in my future classes on gene interaction. That what Mendel actually hypothesized, that each character is governed by a single gene, is not true. There may be multiple genes involved to govern a single character. In that case, the gene symbolizing is really difficult because you will have to first establish whether the two factors which are interacting to produce a particular phenotype are two different genes or two different alleles of the same gene. So this will be dealt by me in my next lecture, that how we can assign allelism through complementation test, that if two traits they complement, then they are non-allelic, but if they fail to complement, then they are possibly the same alleles of the two genes. So if you come to know that they are the same genes involved, it actually happens in case of biochemical pathways that leads to the production of different products in different states. Suppose I give you an example, that you start with substrate 1, which is white in color. One gene acts on it to give a substrate 2, which is yellow in color. Then another product you get, which is pink in color. This reaction is carried over by gene 2. And finally, you get red color product by the action of gene 3. Now think, if you don't have functional gene 3, then you are going to get a pink color product because gene 1 and gene 2 is working. Now, if you don't have both gene 2 and gene 3, you are going to get a yellow product. And if you are going to lose all the three genes, gene 1, gene 2 and gene 3, then and then only you are going to get a white colored product. So if I think that this color is of a flower, so the flower color trait is generally being governed by three genes, G1, G2 and G3, which converts white product to gradually yellow, pink and red. In these cases, you will have to first identify that what are the allelic functions of G1, what are the allelic versions of G2 and what are the allelic versions of G3. Then and then only you can assign their symbols. So thank you for listening. In my next classes, I will be discussing on gene interactions, how different genes interact to produce a particular character and trait. You can post me any questions regarding my presentation at my email ID, that is tirthartha@gmail.com. Let me write it down for you. It is tirthartha@gmail.com. Thank you very much for your listening.